Welcome back, students. Today I am going to share types of stains and techniques used in simple staining. Let's get started. There are three stains are used in microbiology laboratory. First one is simple stains, differential stains and structural stains. Let me explain one by one. Simple stains are the stains which are used in microbiology laboratory for the staining of the microbes and to study the shape and arrangement of cells. Simple staining technique can be defined as coloration of microorganisms by applying single dye to a fixed smear. There are three different types of simple stains are there. First one is basic dyes, acidic dyes and indifferent dyes. Second one is differential stains. This is particularly used for the separation of microbes into groups. Especially this differential staining is used for the differentiation of microbes like gram stains, that is gram positive organism and gram negative organism, or acid fast staining is also used in differential staining. Next one is structural stains for visualizing different structures of cells. For example, cell wall stains, we can view cell wall or the structure of cell wall. Spore staining is there. Capsule staining, you can see the capsule, presence of capsule, and flagella stain, whether flagella is present or no, and nuclear stain. These are the different types of structural stains. Let me explain in detail what is the technique practiced in simple stain. We have to take a clean glass slide for this purpose, and we must take the loopful of inoculum, and that inoculum should be spread a thin film on the slide. After smear preparation, apply about 5 drops of the stain. We can use different stains for this purpose. When you use separate stains, each stain time duration is different. For example, if you use methylene blue, you have to maintain 50 to 120 seconds. If you use crystal violet, then 2 to 60 seconds are needed. If carbon is used, then 10 to 15 seconds are enough. So here we have to maintain time according to the stain which you are using. After applying stain, you have to allow that stain to remain for some time. That is according to the stain which you are using. After that particular time, pour off the excess stain and wash the slide. After time maintenance, you have to remove the stain and wash it under tap water. After washing, you must air dry the slide by using a blotting paper, and you have to air dry it. Then examine the slide under microscope. When you observe under microscope, microbes stain deep blue. When you are using methylene blue, microbes will appear blue color. If there are endospores within the cell, they appear as clear areas. The cells appear deep blue, and if the endospores are present in the cell, then they appear clear areas. Whatever the staining technique which I have explained now is called as positive staining, because in simple staining there are two different types of staining techniques. First one is positive staining, as I have explained now. Another one is negative staining. In positive staining, when you are applying the stain, the microbes will take that stain and background will be colorless or transparent. But in the negative staining, the background will take the stain and the organism appear transparent. Hence, this is called as negative staining. Here we use acidic dye for staining, and these acidic dyes stains only the background, not the organism, and hence the organism look transparent against a dark background. Let me explain what are the different types of simple stains used in microbiology. So simple stain, there are different types. As I said you, that is basic stain, acidic stains and negative stains. The first we will study what is basic stains. There are different specific dyes are used for this basic stain. Methylene blue, crystal violet, malachite green, basic phosgene, carbon phosgene and safranin. These are the specific dyes which are used for basic stains. And what is the purpose of this basic stain? Stain negatively charged molecules and structures such as nucleic acids and proteins. The outcome is, this is the positive staining technique. So this is the picture which you can observe the positive staining. The organisms took stain and background is colorless. Next one is acidic stain. The examples for acidic stains are eosin, acid phosgene, rose bengal, Congo red. And what is the purpose of this acidic stain? Stain positively charged molecules and structures such as proteins. The molecules which are positive in nature, they can be stained by using acidic stain. And outcome of this acidic stain method is, can be either a positive or negative stain depending on the cell's chemistry. It can be a positive stain or can be a negative stain. It depends mainly on the cell's chemistry. This picture will show you how the organisms can take the acidic stains. Last one is negative stains. The specific dyes which you are using for negative stain is India ink and Negrosin. Purpose of this stain method is stains background, not specimen. As I said you in the last session slide itself, that here whatever the stain which you are applying, those will not be taken by the microorganisms, but background will be stained by this negative staining. That's why it is called as negative staining. Outcome of this negative stain is dark background with light specimen. See here, you can easily view this picture, that is organisms looks like transparent and the background is colored one, or background is dark color, dark one. So these are the different types of stains which we can employ in simple staining. Thank you.